In this video, we will discuss about this sting agonist when combined with BH3 mimetics can trigger apoptosis in vivo. So far in the previous videos, we have understand the results that are presented in the paper that sting agonist when combined with these BH3 mimetics, it will trigger apoptosis in the tumor cells. So they wanted to check whether these in vitro results are replicable in the in vivo studies or not. So first they presented their results, in vitro results in the panel A, B, C, D. So we will discuss those in vitro results. So about this combined therapy. So they checked their results in this control cells TRP53 knockout and this buck knockout, which is the apoptosis target genes. So what they have found out is that when they use the combined therapy, that is sting and BH3 mimetics, they see reductions in the tumor cells, and that we can see in all the cases. For this AH15A control cells, we see the reductions in the tumor cells when the combined therapies is used, that is when the BH3 mimetics and the different reagents for this sting agonist are used. So in all the cases, we see reductions in the live cells, tumor live cells, for the control case and also for the TRP53 knockout case. But we do not see any reductions or significant reductions for these buck knockouts. This indicates that it's the apoptosis that triggers the death of the tumor cells. As when the apoptosis target genes are knocked down, they do not see any cell death. And similar results they have seen for these AF47A tumor cells. And here also we do not see any cell death where the buck knockout is cells are used. Thereafter they check the results for this. Then they compare the results between TRP53 knockout and these control cells. And what they have find out is that in the case of this combined therapy, that is as when they use the BH3 mimetics and sting agonist, they see more reductions in the live cells compared to the TRP53 knockout only cells. So this indicates that the combined therapy has a synergetic effect that leads to the further reductions in the tumor cells. And the similar results they have seen, that is the results that they have observed for the AH15A and AF47A, the similar results they have seen for this DHL cell line, that is 214 DHL and 270 DHL cell line. That is when they use the combined therapy, they see maximum reductions in the live cells. And this is what they have finally understood about the mechanism after they did their in vitro studies. That is, when the BH3 mimetics are introduced, they will lead to the inhibition of the pro-survival cells, and this will cause upregulations of these pro-apoptotic proteins, and it will trigger apoptosis. And these things are usually upregulated or triggered P53 to further accelerated these pathways. But in the absence of P53, the roles of P53 can be taken care by sting pathways by uh, triggering sting pathways or activating sting pathways using sting agonist. So this will cause this BH3 only proteins, and this will further accelerate this apoptosis of the cells. So now they wanted to check their mechanism using the in vivo studies. So they use these AH15A cell lines, and which is devoid of the immune cells such as T cells. And then they use this diazoles and this S63845 as a mean of combined therapy. That is, for the sting agonist they use diazole, and for BH3 mimetics they use S63845, and they used at different combinations. That is, they used either diazole or either S63845 or nothing in the control cells and TRP53 cells, TRP53 knockout mice or in the control mice. And what are the results are shown in this panel F and G. So for the control cells, what they have seen is that when they use the diazole only, there is a significant increase in the survival cells, and when they use the this BH3 mimetics only, they see further increase in the cells. But when they use the combined therapy, that is BH3 mimetics and diazole BH3 mimetics and sting agonist, they see a maximum survivals of the cells in the control cells. And when they did the experiment in this TRP53 knockout cells, they see a similar results. That is when they use only diazole, they see a significant increase in the survivable cells. But when they use this BH3 mimetics, they see a further increase in the survivals of the mice, not cells, yeah, mice. And when they use the combined therapy, the survivals of the mice is increased furthermore, though the results are lower compared to the control cells, but the combined therapies results are higher compared to the other therapies in the TRP53 knockout cells. So this indicates, or this results indicates that the combined therapies has a role towards targeting this apoptosis, towards triggering this apoptosis in the in vivo environment on the tumor cells, but it does not have too much impact on the healthy cells. Sting agonist does not have too much impact on the healthy cells or this BH3 mimetics. Or we can say the combined therapies has targeted effect on the tumor cells but not on the healthy cells. This is what the results so far shows. So I hope you understood how this combined therapies has the better results compared to the individual monotherapies. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.